Hello everyone. Thank you Analytika 2022 organizing committee for allowing me to discuss one of my research areas currently undergoing in my research group. Today I am going to discuss one of the most important uh, research areas uh, applicable for the pharmaceutical industry. The topic of my talk is method development, validation, and concentration determination of metformin hydrochloride and atorvastatin calcium using UV visible spectrophotometry. The history behind this project is we were interested to find out a fast, simple, as well as economical way to find out the dosage of a pharmaceutical drug. It's very important for us to have a correct dosage when we administer the drug to a patient. Now, to start with, I would like to give a brief introduction to my uh, talk, and then what is this method that we have implemented in order to achieve our goal. So, and then the importance of the method validation for such studies, and finally the sample analysis uh, demonstrated by this particular method. So these are the steps that I am going to take you through during my talk. So let's look at what is this drug. So as I briefly explained at the very beginning, dosage of a drug is a very important thing. So the drug or the dosage can be correct or wrong. If it is correct, then no problem at all. But if it is wrong, then the consequences are not that good. So if it is a correct dose, then we call it an effective dose. Effective dose means that the person who is suffering from certain diseases or disease can get cured due to the uh, right amount of drug dosage administered to that particular person. But if it is not a correct one, then the problem arises. So then there are two different areas that we need to discuss, that is toxic or lethal dose. If it is a toxic dose, that a person might undergo certain other side effects which might be curable or sometimes they are not curable. Also, the worst case scenario is the lethal dose. So that means the person might get die. Now, the question is, how are we going to know, how are we going to determine the correct dosage? Now, for that, the analytical chemists need to have a particular method developed in order to find out the active pharmaceutical ingredient concentration in a particular tablet, pill, or any other thing that you are going to administer into a patient. Now that part is called the dose formulation analysis as shown here. Now in the dose formulation analysis, what happens is that we have a developed method and we are using this developed method to determine the amount of active pharmaceutical ingredient present in a given tablet. For example, if you take 10 milligram tablet, what is the actual amount of active ingredient? So that's a question mark. And there are a lot of pharmaceutical companies around the world, and they do use different methodologies. And in our case, what we are going to do is we are trying to find out the most simple or the economical method to determine the amount of drug or the amount of active material present in the sample that we are interested in. Now the question is, we need to have a method or a procedure for that. Now how are we going to develop a procedure? So for that, we have another area which is called the method validation and method development, because you can just have an analytical procedure and publish it and say, okay, this is the right way of doing this one. So you can go ahead and do this thing. but that is not practical because we need to do a lot of studies beforehand prior to come to the last step, which is called the dose formulation analysis. Now, not only that, now this method validation or the method development, they are under the surveillance of certain different organizations. So before we go look into that, let's quickly look at what is the meaning of this method validation. It's a process used to confirm that the analytical procedure employed for a specific test is suitable for the intended use. So that's very important. And the results that we obtained out of this one should be able to judge the quality, reliability, and consistency of the analytical result. So once we develop that procedure, it should provide continuously reliable and consistent result, regardless of where you do this test, regardless of what is the laboratory, regardless of the person who is conducting this analytical procedure. To control that, to oversee that, there are several organizations which come up with different rules. Now, three of such organizations and three of the areas that we are interested in our research is ICH, that is International Conference of Harmonization, and the United States Food and Drug Administration, FDA, and Food and Agriculture Organization, FAO. So these organizations present rules and regulations which can be used to develop our drugs or develop the analytical procedure applicable for the drugs. So this is going to be very convenient for the pharmaceutical manufacturers so that they can quickly apply these methods in their laboratories and see whether their drugs are according to that particular guidelines. Now, not only these guidelines, they have some acceptance criteria. So when the drug is developed, the drug should be able to pass all the acceptance criteria. If one or two criteria are failed, then we say that drug is not suitable for the intended purpose. So what is the intended purpose? That is to administer into a patient. Okay, now here we have chosen two drugs to demonstrate our process: Atorvastatin calcium and Metformin hydrochloride. For simplicity, hereafter I will be using Atorvastatin calcium as AC and Metformin hydrochloride as MH. Now let's look at this Atorvastatin hydrochloride. Sorry, Atorvastatin calcium. So it belongs to the group of hydroxymethyl hydroxymethylated coenzyme A or HMG coenzyme A, and commonly known as statin drugs. So we all know about the statin drugs. And these are administered to patients with hypercholesterolemia and the prevention of cardiovascular events. So this is a drug which is really important for people who are having cholesterol and related cardiovascular diseases. And metformin hydrochloride or MH is an anti-hyperglycemic agent used to treat type 2 diabetes. So if you look at the two uh, drugs or the two areas that we have selected, significant percentage of world population is suffering from one of these, either cholesterol or the cardiovascular related disease or diabetes. So therefore I believe this methodology will be really useful for most of the countries to find out whether their drugs which are administered to treat these patients are suitable for the intended purpose. Now these are the two chemical structures which we are going to discuss during the rest of our talk. Right. So our objective is to develop a simple and user friendly. So that's very important, the word user friendly, because any analyst or a trained person should be able to work out this process without much assistance from a senior scientist. So our first goal is to validate according to the guidelines of ICH. So ICH guidelines give all these instructions how to do those things. So we are following that one. In addition that we try to maintain GLP or good laboratory practices throughout our process, because it's very important to ensure the GLP such that our work can be redone without going back to the initial analyst or initial laboratory. And then we want to test whether our method is suitable for the routine quality control analysis of this atorvastatin calcium and metformin hydrochlorides or AC and MH drugs. Okay, so let's look at our plan. So first of all, we have to select the drug standard. Now we have the drugs, we have identified them, AC and MH. Then we need to get the standard. So once we choose the standard, then we need to find out the suitable solvent to dissolve our drug. Since we are going to the simplest method, which we call the UV visible spectrophotometry, which is very simple, which is very commonly available in most of the analytical chemistry laboratories, we need to then determine the wavelength, the absorbance wavelength, or we simply call the lambda max, where the absorbance is highest in that particular range. And then we need to identify the correct wavelength range. Okay, so once we do that, we have to prepare the standard calibration curve, or sometimes we call the working curve. Now this is a very simple way to look at that. So we have a drug now, we are preparing a working curve where our interested concentration is in line. Then we have to check the linearity as well as the other assay parameters. Right. So what are these validation parameters? So that's a question, right? So I will be discussing the validation parameters in one of my next slides. Then finally, we can do the test sample analysis. So that is our plan of work. So let's look at how do we determine the lambda max. So we choose the correct solvent for each of the drugs, and then we scan our drugs using the UV visible spectrophotometry to obtain these curves. Now, if you look at the MH, you can see a peak is coming around here, which is the wavelength of 233 nanometers. So it gives the highest absorbance at 233 nanometers. But if you look at the AC drug or the atorvastatin calcium, you can see it, the spectrum of that one in the dotted curve or the broken line curve, and its highest absorbance is somewhere around 245 nanometers. So therefore, for rest of our studies, for each of these drugs, we have to set our instrument to this particular wavelength whenever we are doing the analysis. Okay, so we have chosen the right solvent for these ones, and for some of the drugs, for some of the samples, we might need to use the co-solvents as well. So it depends on the drug. So when you are doing the analysis, you will understand, okay, whether this is completely soluble. So if it is soluble, then we can use it directly. If it is not soluble, then definitely we need to add certain co-solvent to speed up the solubility or the dissolving process. So this is how we prepare our standard calibration curve. So 10 milligram of the standard sample was weighed. So the standard sample was provided from one of the government agencies in Sri Lanka, because you can't purchase the standard samples from anywhere that you want, because they are not available in a market. And these samples, or the 10 milligrams, are transferred and dissolved in a 100 milliliter volumetric flask with deionized water. So our intention purpose was to prepare 100 ppm sample. Then we prepare a series of solutions from this stock solution. Now our stock solution is 100 ppm, and we prepare the series of samples based on that. Then we measure the adsorbance in our selected wavelengths with 233 or 245 based on depend on the drug. Then we develop the calibration plot, and then linearity and correlation coefficients were determined to see whether we can continue from that one. Let's say that we did not get a linear curve, so that means then we have to think about another avenue to come up with a linearity, because linearity is one of the most important thing to continue with this process. Okay, so these are the method validation parameters. So now in the rest of our talk, we are going to discuss these parameters briefly and how did we achieve this one. Now, not only that one, we need to ensure that all these met the acceptance criteria in order to use that method as a validated method. So linearity, range, LOD, LOQ, precision, accuracy, and stability. Now these are the most commonly used method validation parameters. Regardless of what technique you are using, you got to use these things. So it could be U visible, it could be HPLC, it could be GCMS, right? But the advantage of our method, our UV visible method, is that compared to the chromatographic method, this method is way simpler. Right, now these are our working curves. Once we perform our once we prepared our standards, and once we performed the absorbance for once we measured the absorbance for all of these things, all of the drugs in our range, so we were able to obtain two linear curves for AC and MH. Now as you can see that in both cases their R square values are almost 1. So that means that we are able to obtain a linear curve for this one. And from this one, not only the linearity, we can determine the range as well. Now, if you look at this one, we have the particular range from 2 ppm to 10 ppm for the case of MH. For the case of AC, we have the concentration range from 5 ppm to 15 ppm. Now the question is, why did we select those two only? Why can't we go beyond that? We tested that, and then we found that the linearity is leaving off. So it is not going to be linear. So this is the linear dynamic range for that particular drug. So that's why we selectively chosen this range. Right. So these are our results now. So linearity and range. As you can see, the correlation coefficients are really good. So we accepted, okay, that these two methods that we are implementing for this particular drugs are good for us, good for the intended use. And the concentration limits, we can have two different types: lower concentration limit as well as upper concentration limit. For each drug, I have shown you the lower and the higher or the upper concentration limit. So whenever we are doing our tests, we want to ensure that all our concentrations are in this range, and if they are above these concentrations, then we need to dilute that, and if they are lower in the concentration range or the out of this range, then we have to concentrate to get them back to the right concentration range. So therefore, whenever we do the sample analysis, we wanted to ensure that our concentration should be in or within this range, right? So let's look at the limits. So limit of detection and limit of quantitation are two important parameters that is determined in this methodology, and here we have two equations which we use to calculate that. So those are calculated parameters. So LOD is calculated by 3.3 sigma over S. Now here sigma is the standard deviation of the response. So we can calculate the standard deviation. And the slope of the calibration curve, that is also calculated either from the software or just in other method. So that is the LOD, and LOQ is 10 sigma over S. Now, even though I have just shown you just one calibration curve, it is in fact not just one calibration curve. Now we have done this analysis significant number of times, and using other analyst, not only one analyst, several analysts have done this work and to see whether we can get this linear curve. Now these are summarized or the average results that we are demonstrating. So therefore we have a good standard deviation value for this analysis, and we have enough data to prove that one. And then we come to the repeatability or the precision. So precision is also a very important parameter, and if I briefly explain the precision, it is not really the accuracy, how closeness your readings are to each other. Now here we have demonstrated it by using 6 samples. So we prepare the same concentrated sample 6 times and we get the average of these things, and you can see the standard deviation. This standard for the standard deviation, it's 0.001 for the AC and 0.011 for MH, and the relative standard deviation, this one is very low, this is little bit higher. However, the ICH guidelines give a limit for us. So it says if the percent RSD is less than 2%, then we can accept this as a valid precision. So in either case, this value is less than 2%. So therefore, it passed, the both drugs pass the acceptance criteria for the repeatability. And another way of look at this precision is the intraday precision. Now, when we have this sample stock solutions, we wanted to keep it for a while because we can prepare this uh, drugs each and every day. So we looked at how they vary day by day. For that, we have 3 different concentrations for each drugs. So for the AC, 5, 10, 15, and MH2, 6, 10. So those are the 3 different concentrations that we used, and we measure the absorbance and we calculate the percent RSD values for each of them and found out their percent RSD values are less than 2%. So that means our interday precision is also within our range. So that means even though that we prepare it today, if we measure it tomorrow or some other day, still that we were able to obtain the same or similar results. Now when we come to the accuracy, it's a very complicated one to do the analysis. So what we have done was, for each drug that we have prepared different concentrations, right? So in this case, we have recovery levels of 50, 100 and 150, uh, and based on that we calculate the mean recovery. So here that we have spiked a certain concentration, which is a fixed concentration, to 3 different brands of tablets. So we have chosen our samples beforehand, and then we spiked our sample standards to them, and then we calculated the percent recovered. Now as you can see that the percent RSD values are again less than 2%, and also this recovery level is in between 90% and 110%. So in other words, it is 100% plus or minus 10%. So that means it again meets the uh, acceptance criteria developed by the ICH guidelines, and we did the same thing for our MH drug, and again we found out the our requirements are met there. So we can conclude that our method has a good accuracy to the intended purpose. And then one of the most important parameters is the stability. So no matter how good our process is, if we can't use it for a few days consecutively, so we prepare a set of samples, and if we want to do the analysis tomorrow or some other day later, then if we can't use that standards, then it's going to be a problem for us. So for that, we have determined the stability. So we determined the stability of both AC and MH, and we found out the AC has the stability at room temperature and refrigerated conditions up to 7 days. And in fact, we have tested for 14 days. For some reason, one of the storage uh, is failing after 7 days. So we have done this test several times, but again, that 7 day period is more than enough. So in other words, if you prepare the samples today, the standard or the samples, you can keep it in the room temperature or refrigerated for 7 days until you complete the testing. And as you can see, the percentage difference is very minimum at the room temperature, so means that their decomposition is very low. However, when you come to the refrigerated uh, storage condition for this RF, so one concentration, that is 5 ppm concentration, it is relatively higher. The change of the absorbance or the change in the concentration is relatively higher. Still, it is within the range, so therefore we can use it for the intended purpose. And stability of MH also demonstrate similar curves, and again we found out that the MH is stable at RT conditions as well as RF conditions up to 7 days, or the room temperature conditions and the refrigerated conditions to 7 days. So in conclusion, that we can say both drugs are stable for 7 days at this given in this given storage conditions. Then we came to the analysis of the test samples, because we wanted to test whether our method is good. So for that we have purchased some brands of AC from different pharmacies located in Sri Lanka, and for the AC, approximately they are 10 milligrams. And from our method that we have found out, about 90 to 98.8% range, these active pharmaceutical ingredients are present. So in other words, that the brand tested met the acceptance criteria and hence suitable to administer to the patient. So these drugs are good there. Now we can determine other brands as well and see whether they are in this range. So if the concentration is in this range, or the percent active ingredient concentration is in this range, then they are good for the intended purpose. Once we come to the MH, then we observed some uh, special things. For example, uh, for brand A, the concentration is 82.2%, and brand E, it is 90.8%. So in fact, it is the highest concentration that we have observed for out of all these 6 brands, 90.8%. And if you look at F, it is 79.8%. Now you can't round it up to 80 and say, okay, it is within plus or minus 20%, so that is the ICH guideline, and it is in the usable range. You can't say like that, because it is below that range. So therefore, this this drug F might cause some side effect to the patient. So we can do this kind of analysis and we can conclude whether that drug is good for the intended purpose or not based on our results. So to conclude my talk, now we have developed a very simple method using UV visible spectroscopy. Now there are other methods, as I mentioned in my talk, related to the chromatographic techniques. Now, since the chromatography is so expensive, this is a very user-friendly and economical method also, which follows the ICH guidelines. And our method was tested and it was meeting all the acceptance criteria that the ICH has set to method validation process. And this can be used for the quantitative analysis, and also our samples are stable at room temperature as well as in the refrigerator condition. So I believe that this gave you some insight about a new area that where you can test your samples at your laboratory prior to release them into the market. So with that, I would like to thank the Institute of Chemistry Ceylon for providing the financial support to conduct this research, as well as the State Pharmaceutical Corporation providing us the uh, standard samples through one of the contacts, and also the two research assistants or the two students who worked under my supervision. Currently they are working in different places. Mr. HMMB Herat is a assistant chemist at distilleries company Sri Lanka, and Vishaka Kaushalya, she is currently a graduate student at the Wayne State University, Michigan in the USA. So I must thank them for doing this work. It's not a simple work, it's a very tedious work. Uh, and with that, I would like to conclude my talk. And if you have any questions, I am happy to answer your questions. Thank you very much.